Humans have 46 chromosomes found in each of their cells. There are, however, only 23 unique chromosomes since one set of 23 is inherited from each parent. Paternal chromosomes are inherited from the father. Maternal are inherited from the mother. Since these chromosomes contain the same genes, with just slight variations in the sequence of DNA, they are called homologous chromosomes. The cells that contain 46 chromosomes are called diploid cells, but to form reproductive cells, homologous chromosomes must be combined so that the cell has only 23 chromosomes. In other words, the cell's ploidy level must be reduced from diploid to haploid. This task is accomplished through meiosis. Like mitosis, meiosis is preceded by DNA replication. Each chromosome is duplicated so that it consists of two sister chromatids. At this point, the DNA is found in an uncondensed form called chromatin. Prophase 1 is the longest phase, taking up to 90% of meiosis. As in mitosis, the nuclear envelope breaks down and chromosomes condense. Chromosomes line up with their homologue and become bonded by a protein called the synaptonemal complex, preparing them for crossing over. This bonded group of 4 chromatids is known as a tetrad. Homologous chromosomes begin swapping corresponding sections of DNA. After the crossing over happens, small X-shaped regions called chiasmata form where DNA was exchanged. Crossing over is a major contributor to genetic diversity because it creates new combinations of genes. After crossing over, the spindle fibers attach to the sister chromatid's kinetochores to prepare them for metaphase 1. As metaphase begins, chromosomes line up along the metaphase plate. Random positioning of the maternal-paternal chromosomes also contributes to genetic diversity. In anaphase 1, the synaptonemal complex between homologous chromosomes is broken, and motor proteins begin transporting the separated sister chromatid pairs along the spindle fibers to the opposite poles of the cell. In telophase, the nuclear envelope reforms and cytokinesis commences. Cytokinesis divides the parent cell into 2 daughter cells. These individual cells can now enter meiosis II to produce gametes. Meiosis II is essentially identical to mitosis, except a cell entering mitosis is diploid, whereas a cell entering meiosis II is haploid because chromosome duplication does not occur before meiosis II. Prophase II begins as the centrosome duplicates and migrates to the poles of the cell. The nuclear envelope dissolves and the spindle fibers form and begin to attach to the kinetochores of sister chromatids, jostling them into position. During metaphase 2, the chromosomes are positioned at the metaphase plate. Because of crossing over, the 2 sister chromatids composing each chromosome are not genetically identical. When the cell enters anaphase 2, the kinetochores break down, allowing the chromatids to pull apart and become individual chromosomes. These genetically different chromosomes are transported to opposite poles of the cell. In telophase, the nuclei reform and the chromosomes decondense. After cytokinesis, the single parent cell that entered meiosis I is now 4 haploid daughter cells, all with differing DNA. These reproductive cells that are created as products of meiosis are called gametes.